I'm Madelyn Schider, and I will be presenting my research on the effect of micronutrient levels of iron, manganese, molybdenum, and zinc on Catawba grapes, scientifically known as Vitis vinifera. The main reason I chose to conduct this research was because in the future I would like to pursue a career in owning a vineyard. I wanted to work with grapes in some way, and so I narrowed down the necessities of vineyards. The main requirements for succeeding in growing grapes come down to the climate, which fluctuates where you are and is vital in certain species of grape, water availability, which is not as much as one may think, and of course, soil composition. After deciding to focus on soil, the four major differences in soil to look at were mycorrhizal fungi, pH, macronutrients, and micronutrients. For further background knowledge on macronutrients and micronutrients, macronutrients are used in large quantities in plants. The elements that are considered macronutrients in plants are nitrogen, phosphorus, and potassium. These elements are vital in the growth and development of plants. Micronutrients, on the other hand, can be as little as 2% of what the plant is actually taking in. These are elements used in small quantities but are essential for healthy development. As they are used in small quantities, if there is too much or too little, development can be disrupted completely. The picture to the right is the research I went off of. Their focus was on geraniums, and as you can see, there came a point where the concentration levels became toxic to the plant. Now going into my focal nutrients. These four were chosen since there was little past research done. Why each of these are important in development is iron is vital in assimilation, respiration, and chlorophyll formation. Manganese is vital in chlorophyll synthesis and nitrogen metabolism. Molybdenum is fundamental in nitrogen metabolism and growth. And zinc is important for protein synthesis, hormone production, and fruit production. All of these have two things in common: photosynthesis and growth. Chlorophyll is the primary pigment used in photosynthesis and is what gives plants their green color. In my research, I found the chlorophyll A and B ratio, which is usually a 3-to-1 ratio and is considered when seeing a stress response in plants. A major concern was to acquire a grape that would work in the bipolar Michigan climate. With further research, Dr. Heckel and I found AA Vineyard, which is located in New York. This vineyard grows and sells vine cuttings, which can be easily planted and grown. We chose to work with the Catawba grape, which is usually used in sparkling wine, juice, and jellies. The main reason for using this grape is because it is a very hardy grape and can withstand fluctuation in temperature and humidity. Going into my hypothesis, I thought with an increase in concentrations, there will be an increase in growth, higher chlorophyll A to B ratio, increase in biomass, and evidence of toxicity in the higher concentrations, meaning once toxicity was reached, all of the above would substantially decrease. The planting process, in short, was to take the vine cuttings seen on the left and do the same process for each one. I started with a neutral peat moss soil placed in a grow bag, added the vine, covered the roots with more peat moss, and then finished it off with potting soil. This potting soil had a set concentration of macronutrients added in order for proper development. Lastly, they were all watered with distilled water. To keep track of all these plants, 6 pallets were set up in the Strohsacker greenhouse. Plants were placed in a random assortment, and the surrounding area was sprayed with a fungal pesticide. Also, the leaf count and height were measured to have a starting point for each vine. In order to get all the micronutrients into the soil, I mixed 20 jugs of distilled water with the needed nutrients in order to get an aqueous solution. The nutrients added were iron sulfate, magnesium sulfate, sodium molybdate, zinc sulfate. Once mixed, the 7 replications of each group were watered 3 times a week with 750 milliliters of the nutrient solution. The 5 controls were also watered with regular distilled water the same days. This went on for about 6 weeks. The measurements found for the plants were height, leaf count, leaf width, biomass, and the frozen leaves were used for chlorophyll analysis. In order to see if the soil actually did take up nutrients, the soil was sent to the lab to test for micronutrient levels. To get the biomass, all the greenery of each plant was cut off and stuffed into paper bags. The vines were then placed in a dry oven for 24 hours at 120 degrees Celsius in order for all wet mass to diminish. The bags were then weighed and the mass was calculated. To prepare for chlorophyll analysis, the first replicate of each treatment group was ground with a mortar and pestle, and liquid nitrogen was added in order to grind smoothly. The powder was then placed in tubes with 80% acetone, left to soak, and then centrifuged at 13,000 rates per minute for 15 minutes. The aqueous solution was then separated and put in cuvettes in order to be analyzed by the spectrophotometer to find the absorbed light at the wavelengths of 470, 650, 646.8, and 663.2 nanometers. Once these levels were found, the ratio for chlorophyll A and B were found. Finally, going into the results, all p-values above the bar graphs in the next 4 slides are based on the significant difference of the groups and the controls. Amazingly, the control vines, when it comes to height, were 30 to 50 centimeters longer than most of the treatment groups. The controls were all significantly higher than the treatment groups other than zinc. The treatment groups alone did not have any significant differences, but it is interesting to see that the controls exceeded expectations. The most significant result in height was iron, where shown in the graph, the highest concentration is only half as tall as the 1 millimole concentration. This trend, as you can see, slowly increases and dramatically decreases, which could be a sign of toxicity. Going into leaf count, as you can see on the controls, there were twice as many leaves as the treatment groups. There were no significance was found for leaf count in the comparison of treatment groups, but controls again outperformed the concentrations completely. Similarly to leaf count, the mass of the controls exceeded expectations and were more than double the weight of the treatment groups. The most interesting trend of the leaf count within treatment groups was manganese, which shows a trend of slow increase and a dramatic decrease by the highest concentration. If you look at the bar graph, this on a larger scale would be evidence of toxicity within that treatment group, but the concentrations, yet again, were not high enough, as we'll see later. The controls in the leaf width were slightly larger except for zinc, which raises curiosity. Zinc was at a mere 12.5 centimeters, while the other leaves were averaging a width of close to 17 centimeters. This may be because zinc is not vital in chlorophyll development, and its focus went toward the vines themselves, or in some cases, the production of fruit. Seen here is a graph of soil composition. This graph shows the different concentrations within each treatment group and the levels of that nutrient found within the soil. The rating is based on optimal levels for crops. This explains why there was no significance in most of the treatment groups themselves. As you can see, the soil did not absorb the adequate amounts of the concentration we wanted. Iron, for instance, only had low ratings for every treatment group. Manganese reached an acceptable amount by the last concentration. Zinc was the only group that reached an optimum level of concentration, but interestingly, the controls all had low levels as well, meaning they did too not have adequate amounts of these micronutrients, but still outperformed most groups. This gives solid evidence that micronutrients may not play a vital role in these growth measurements. Analyzing the chlorophyll levels was interesting. As I said earlier, 2 of our nutrients, iron and manganese, would hypothetically show different results. By looking at the graph, you can see that iron did increase in chlorophyll A to B ratio, but manganese dropped at the second concentration, similarly to molybdenum. Zinc, on the other hand, had an interesting hump where this could be the beginning of a toxicity level. Simply looking at the physical characteristics of iron compared to the other treatments, there was a yellow blotching effect on most leaves. The stems started to have a darker red hue as concentration increased, and there was evidence of stunted growth and drying out of tendrils. To the right, you can see is an example of a tendril. The physical manganese characteristics were that there was a strong green with stark red vines through all concentrations. The soil composition was acceptable in the highest concentration, but there was also still no significance in biomass, height, or chlorophyll production within the treatment groups themselves, though the controls did outperform in most categories. For the physical molybdenum characteristics, the lower concentrations had minor signs of wilting. There was healthy growth in girth and leaf width as seen in the picture. The higher concentrations also had signs of stunted growth and discoloration, and there was no information on soil composition, sadly, so we have no idea how much molybdenum was actually in our soil. The physical zinc characteristics were that the soil composition did reach an optimum level, which was our only one. Chlorophyll production also looked interesting, but there was not sufficient data to compare significance. The leaves were also comparatively smaller than other treatment groups. which could also be another research project to do. In conclusion, there were no significant differences between treatment groups themselves, meaning micronutrients did not play a role in height, leaf count, biomass, or the chlorophyll A and B ratio. But in some cases, it did seem that toxicity was close to being reached. The variables that could have affected these results were the temperature, humidity, but most importantly, the space given to the grapes to grow. Also, the results were swayed by the fact of the concentration levels not being high enough in the soil, meaning the plants were never at a toxic level of micronutrients. Lastly, the coolest phenomena that took out of this research was that one of my vines actually began to produce fruit. This was, of course, in the highest concentration of zinc, which, as stated before, helps in the development of fruit. As you can see in the picture, these are the buds of grapes beginning to form. For future research, I would recommend a focus on one treatment group, as 4 different micronutrients became overwhelming. I would also recommend growing grapes in a natural setting. Grapes prefer to grow about 10 feet away from other grapes in order to reach maturity. I would also definitely use higher concentrations of micronutrients since the soil did not show adequate levels. I would also increase the sample size for a higher chance of significance. And lastly, I would test the soil for all nutrients, not just the one being tested. This could show possible cross-contamination from the other treatment groups. I would like to thank all these people for funding my research and making it happen. Here are my references. Thank you for listening.